So I'm just going to expand on Marguerite's overview of vitamin C by talking more specifically about the human intervention studies we've carried out around the health and immune benefit functions of kiwifruit-derived vitamin C. And so I'll start with just talking a bit about some of the bioavailability studies we've done, where we've specifically looked at uptake of vitamin C from kiwi fruit. And then I'll talk a bit more about some of our more recent functional studies, where we were particularly interested in the effect of kiwifruit-derived vitamin C on well-being and immune function. And then I'll just wrap up with a little bit of a discussion around the recommended dietary intake of vitamin C. So this table shows you the vitamin C content of different foods. You can see that not all foods are created equal with respect to their vitamin C content, and we heard a similar message yesterday with respect to their sugar and fibre content as well. Most of us are familiar with citrus fruit being quite high in vitamin C, so oranges, for example. However, a lot of commonly consumed foods such as apples and bananas are quite low in vitamin C content. And I've highlighted kiwi fruit because not only are they an exceptional source of vitamin C, but they're also the primary food source that we've been using for our intervention studies. So we've carried out 3 intervention studies, and I'll talk about a couple of them today. The first was a pilot study to determine the dose of kiwi fruit for optimal vitamin C uptake into circulation. And the second was our main bioavailability study, where we built on the pilot study by also looking at uptake of vitamin C into tissues and white blood cells. So all of our intervention studies have been carried out at Primoris Clinical Trials Unit in Christchurch, and this unit comprised a 10-bed ward, which meant that we could get 10 participants in at any one time, and this worked out really well for taking tissue biopsies, for example, because the surgeon could just walk from one bed to the next and collect the muscle tissue. So in this pilot study, it was designed to assess vitamin C uptake from kiwi fruit into plasma and also urinary excretion. And in this study, we supplemented 15 young men with half to 3 kiwi fruit a day for 4 to 6 weeks each. And this was followed by a washout period where the kiwi fruit was removed from the diet. The lead-in period allowed the participants time to lower and control their vitamin C intake prior to taking the kiwi fruit intervention. Blood samples were taken every week throughout the duration of the study, and other samples were collected at baseline and then at the change of every intervention and at the end of the washout period. So if we look at uptake of vitamin C into the plasma, what we found was that as little as half a kiwi fruit was sufficient to provide a significant increase in the participants' plasma vitamin C levels, up to 50 micromolar, which is what we consider to be an adequate level. However, you can see that an intake of 2 kiwi fruit was required to provide saturating levels of vitamin C, and that corresponds to an intake of about 200 milligrams of vitamin C. And we know this is a saturating level because at that intake of 2 kiwi fruit a day, you can see that you get an increased excretion of vitamin C in the urine. You can also see that when the intervention is removed from the diet in the washout period, both the plasma and the urinary vitamin C levels return to baseline. And this drop back to baseline shows how important it is to maintain a regular intake of vitamin C in the body. So our main bioavailability study comprised two arms. The first arm was a low-dose intervention group, which the participants received half a kiwi fruit a day for 6 weeks. The other arm was a high-dose intervention group where the participants received 2 kiwi fruit a day for 6 weeks, and this was followed by a washout period. And once again, fasting blood samples were taken every week throughout the duration of the study, and other samples were collected at baseline, at the end of the intervention, and then at the end of the washout period. So So this slide shows our study protocol, whereby we screened 134 young men for eligibility in the study, and that included having a plasma vitamin C level less than 50 micromolar, so less than adequate. The importance of that is that if the participants already have sufficient vitamin C levels, you're not going to see an effective intervention. So it's important to choose your study cohort carefully and make sure they have low vitamin C levels to start with. And so 36 of these participants were randomly assigned to either the low dose group, who received half kiwifruit, or the high dose group, who received 2 kiwifruit. And so it meant we had 18 participants in each group. And the measurements we assessed were dietary vitamin C intake, and we did that using food and beverage diaries. And we also measured vitamin C levels in plasma, urine, semen, leukocytes or white blood cells, and muscle tissue. And the vitamin C levels were measured using high-performance liquid chromatography with electrochemical detection, and that is the gold standard for measuring vitamin C levels. So I've mentioned that vitamin C levels plasma levels of 50 micromolar and above are adequate. 70 micromolar and above are saturating. Levels below 23 micromolar are considered marginal. And what you can see from this graph is that if a person with marginal vitamin C status ingests half a kiwifruit a day, it takes at least 4 weeks for their plasma vitamin C levels to reach maximum, and that amount does not get above 50 micromolar, so barely adequate. However, if they take 2 kiwifruit a day, their plasma vitamin C levels reach saturating within a week, and those saturating levels are maintained until the intervention, or the kiwifruit, is removed from their diet, after which their levels rapidly drop back down to baseline. And once again, this rapid drop shows how important it is to maintain a regular intake of vitamin C in your diet. This figure shows vitamin C levels in skeletal muscle biopsies. And you can see that there's a linear uptake of vitamin C into muscle tissue with increasing plasma levels. And you can see that saturating plasma levels give you the best muscle status. And we also saw similar effects with white blood cells. We saw a linear uptake of vitamin C with the bigger increase in dietary intake and plasma levels of vitamin C. So what have we learnt from our bioavailability studies to date? We've been able to determine what we would consider an optimal intake of kiwifruit. We found that half a kiwifruit a day, which provides an intake of 50 milligrams of vitamin C, will give you adequate plasma levels. However, an intake of 2 kiwifruit a day, which gives you 200 milligrams of vitamin C, will give you a saturating, or what we would call an optimal vitamin C status in your blood. We also looked at tissue and cell uptake of vitamin C, and we found a linear uptake of kiwifruit-derived vitamin C into skeletal muscle tissue and white blood cells. So in other words, the more kiwifruit you eat, the better your muscle and cell levels will be. So I guess the question then is it important to have saturating plasma status and optimal tissue and cell levels of vitamin C? So as Margaret has already mentioned, vitamin C has a number of important functions in the body because it's a cofactor for a large family of these metalloenzymes which have numerous regulatory and biosynthetic roles. And I'll just quickly go through these again. So these enzymes are involved in collagen tertiary structure, mitochondrial energy production, hormone and neurotransmitter synthesis, metabolic regulation through control of that HIF transcription factor, and also epigenetic regulation through control of DNA and histone modifications. And so what does this mean? Well, it means if you give someone vitamin C, someone who has less than adequate levels of vitamin C, it is likely to be having a beneficial functional effect in the body. So as far as our functional studies go, we were particularly interested in the effect of kiwifruit-derived vitamin C on measures of well-being such as vigour. And we are also interested in the effect of kiwifruit-derived vitamin C on immune function, in particular leukocyte function. So with respect to the effect of kiwifruit-derived vitamin C on well-being, it's interesting to note that the symptoms of depression, fatigue and irritability precede the overt physical symptoms of the vitamin C deficiency disease scurvy. So this indicates an important role for vitamin C in general mood and well-being. And as Margaret mentioned, the brain has some of the highest levels of vitamin C in the body, and it's also one of the last organs to deplete during deficiency. So this suggests a very important role for vitamin C in brain function. So in this study, 35 young men were supplemented with either half or 2 kiwifruit a day for 6 weeks, and their general mood was assessed before and after the intervention using the POMS, or Profile of Mood States questionnaire. This questionnaire assesses tension, depression, anger, fatigue, confusion, and vigour, and comes up with what's called a total mood disturbance score, which is the sum of the first 5 minus the vigour score. And what we found was that the group who consumed 2 kiwifruit a day had a 35% decrease in their total mood disturbance, which was trending towards significance. But you have to realise that this group of participants was recruited based on their vitamin C status, not on their mood status. So if we looked at the subgroup of individuals who had high mood disturbance at study entrance, we then saw a significant decrease in fatigue, a significant increase in vigour, and a 38% decrease in their total mood disturbance following supplementation with 2 kiwifruit a day. And it's interesting to note that no effect of half a kiwifruit a day was observed. So this suggests a dose-dependent effect where it's likely that you need the higher intake of 2 kiwifruit a day to notice these beneficial effects on well-being. But we need to do larger studies to confirm that. So what are the potential mechanisms by which vitamin C could be enhancing well-being? Margaret's already mentioned that vitamin C is a cofactor for the 2 hydroxylating enzymes required for the synthesis of carnitine. And carnitine is the molecule that carries fatty acids into the mitochondria for production of metabolic energy. So this is a plausible mechanism by which vitamin C could potentially decrease fatigue and increase vigour. Vitamin C is also a cofactor for enzymes involved in neurotransmitter synthesis. It's a cofactor for the enzyme dopamine hydroxylase, which converts dopamine into norepinephrine. And it also stimulates the rate-limiting enzyme tyrosine hydroxylase through recycling its enzyme cofactor tetrahydrobiopterin. And a similar mechanism to this has been proposed for the conversion of tryptophan into serotonin. And both of these neurotransmitters have a lot of roles in the body around energy and mood. So this is another potential mechanism by which vitamin C could improve feelings of well-being. And as I mentioned, we're also interested in the effects of vitamin C on immune function, in particular the function of white blood cells. And as Margaret mentioned, neutrophils are our primary line of defence against invading pathogens. And what these cells do is they migrate out of the bloodstream into the tissues and move towards sites of infection where they engulf the bacteria and kill them. Neutrophils have a number of different mechanisms by which they can kill bacteria. The engulfed bacteria are exposed to a number of compounds, including highly reactive oxygen species and proteolytic enzymes, which kill and digest the bacteria. And recent research has also shown that neutrophils can spit out what are known as neutrophil extracellular traps, which trap bacteria and can also kill them. And following bacterial killing, the neutrophils undergo this process of programmed cell death, or apoptosis, which allows them to be engulfed by macrophages and cleared from sites of infection without damaging host tissue. And Margaret's already mentioned that her animal studies have shown that vitamin C enhances this process of apoptosis and clearance by macrophages. So we carried out a human study to investigate the effects of kiwifruit-derived vitamin C on neutrophil function. In this study, 12 young men were supplemented with 2 sun-gold kiwifruit a day for 4 weeks, and their fasting plasma vitamin C levels were assessed every week, and neutrophil vitamin C levels and other functional aspects of neutrophils were assessed at baseline and then following following the supplementation. And the assays we measured with respect to neutrophil function were chemotaxis or migration, oxidant production, extracellular trap generation, and apoptosis. So as we had seen previously, supplementation with 2 kiwifruit a day significantly increased plasma vitamin C levels, and we also observed a significant increase in neutrophil vitamin C levels following supplementation with 2 kiwifruit a day. Furthermore, we saw an increase in neutrophil chemotaxis or migration and an increase in neutrophil oxidant production. So both the increased chemotaxis and increased oxidant production would likely translate into improved immune function, although larger studies would need to be done to confirm these findings. Because kiwifruit contain a number of micronutrients in addition to vitamin C, it's not possible to rule out an effect of these other micronutrients on mood and immune function. But if we look at which nutrients increase the most following supplementation with 2 kiwifruit a day, you can see that vitamin C is by far the most outstanding and therefore the most likely to be having functional effects, particularly in light of its known mechanisms of action. So what have we found with our functional studies? We found that 2 kiwifruit a day enhances well-being by decreasing fatigue and increasing vigour. We have found that 2 kiwifruit a day enhances neutrophil vitamin C levels and also specific functions such as increased chemotaxis and increased oxidant production. And both of these would likely contribute to enhanced immune function. So if we look at the recommended dietary intake for vitamin C, you can see that in New Zealand we currently have one of the lowest recommended intakes in the developed world. And the reason for this is the different criteria that are used to derive the intakes of vitamin C. So in the UK, Australia, New Zealand, prevention of the deficiency disease scurvy is the primary criteria used to derive their RDI. However, in other countries such as North America, potential health effects such as antioxidant protection are used to derive their RDI. And just recently in Europe, their RDI was increased to reflect an intake that would provide plasma levels of 50 micromolar, and that's what we consider adequate, and that's probably the best criteria to use at this stage. Oops. So is it time for a new RDI for vitamin C? Levels below 11 micromolar vitamin C in the plasma are considered deficient and at risk of developing scurvy. Levels below 23 micromolar are considered marginal and prone to becoming deficient. And you can see that intakes of 100 to 200 milligrams a day are required for adequate to saturating plasma levels, so 50 to 70 micromolar levels in your plasma. So in other words, if we give a deficient person the current New Zealand RDI for vitamin C, their plasma levels would still be well less than 23 micromolar, so very marginal. In the early 2000s, the US RDI for vitamin C was increased from 60 to 90 milligrams a day, and so that provides closer to what we would call an adequate level of vitamin C. And as Margaret has mentioned, animals, at the top of the figure, always maintain animals which can synthesise their own vitamin C, always maintain their plasma levels at saturating point. And so we think it is time for New Zealand to increase the RDI for vitamin C, and we'd recommend an intake of 200 milligrams a day because we've shown that this provides optimal plasma and tissue levels of the vitamin. And this recommendation is supported by the New Zealand Ministry of Health, with their suggested dietary target for the reduction of chronic disease risk being 200 milligrams a day of vitamin C. This amount of vitamin C can be obtained from a diet of 5 to 7 fruit and vegetables a day, provided that at least one of them is a high vitamin C food, such as kiwi fruit. So I'd just like to thank the other members of the vitamin C team and acknowledge our primary funders, Zespri and MB. Thank you for your attention.